Hi guys, Miss Francis here today to talk to you about the varied functions of membrane proteins. Well, first of all, let's address the fact that the currently accepted theory is that membranes are a fluid mosaic. Fluid meaning that they're flexible, they're dynamic, not static. And mosaic, meaning that there are many different components that come together to form this cell membrane, this plasma membrane. So the first component that we're going to talk about are the membrane proteins. And those membrane proteins themselves differ in structure and function. There are two types of membrane proteins, and those types are based upon their location within the plasma membrane. So peripheral proteins are located outside of the cell membrane, on top of the cell membrane, and integral proteins are located throughout the plasma membrane. Let me get into greater details. So peripheral membrane proteins are not embedded in the lipid bilayer. Instead, they can rest, or they're loosely bound to the surface of the membrane, or they can be bound or connected to other membrane proteins, meaning the integral proteins. So let's discuss a little bit more about the integral membrane proteins. They are transmembrane proteins. Sometimes they are known as intrinsic proteins, where they are in contact with both the inside and outside of the cell. So there'll be regions that are in touch with the extracellular fluid, regions that are in touch with the phospholipid tails, and regions that are in contact with the cytosol, where the core of the lipid bilayer would be in contact with the hydrophobic regions of the membrane protein, and those are the hydrophobic regions of that member protein with nonpolar amino acids usually coiled into alpha helices. On the other hand, outside and inside of the plasma membrane, you'll have the hydrophilic part of the plasma protein, where there are regions of polar or charged amino acids. Now, despite having these two different types, membranes will also have two different faces. In other words, I mean that the side facing the extracellular fluid will differ from the side facing the cytosol, and it will differ in both lipid and protein composition. Furthermore, the outside will have carbohydrates. So that's how they differ in structure, but they also have a great variety of functions. So as far as transport goes, my plasma proteins could potentially be carrier proteins. They could be channel proteins. Maybe they are pump proteins. Needless to say, they're all going to help transport materials inside and out of the plasma membrane. As far as the proteins that enable enzymatic activity, obviously they're going to play a role in chemical reactions. There are some proteins that play a role in signal transduction, where specific molecules are going to bind to receptors that are ultimately going to send some sort of signal. It's going to trigger some sort of reaction inside of the cell. There are membrane proteins that attach to the cytoskeleton and provide anchorage or stability for the plasma membrane. There are also the carbohydrates that will be found on the surface of these membrane proteins, and they're responsible for cell-to-cell recognition. They're responsible for cell-to-cell communication. So how does one cell recognize another cell? These carbohydrates are called oligosaccharides, and taken together, the oligosaccharide along with the membrane protein, that's known as a glycoprotein. Lastly, you've got membrane proteins that are going to function in intercellular joining. These are some varied functions of your membrane proteins. So here's a little elaboration about the membrane proteins that reinforce the shape of a cell and provide a strong framework. That's by those membrane proteins binding to the cytoskeleton, both on the cytoplasmic side and the exterior side. It's going to help anchor that cell membrane. And here's a little bit more detail regarding the carbohydrates. Like we said, they're oligosaccharides that are on the surface of the membrane that distinguish one type of neighboring cell from another. Well, why would this be important? This is important because I want to distinguish from cells that I need versus cells that I don't need. For example, I need to identify which cells are foreign, which cells are pathogenic that my body would need to destroy and get rid of. So that's it for membrane proteins. Next time we're going to discuss how you could draw a fluid mosaic model.